My name is Ruth Ballard. I'm the Chair of Biological Sciences, and I want to welcome you to the University, to Sacramento State tonight, and our Topics in Regenerative Medicine series. Tonight, Dr. Brian Jonas is with us, and he'll speak about leukemia stem cells targeting acute myeloid leukemia's Achilles heel. Sounds super interesting. I can't wait to hear what's going on in this field right now. This talk is part of a lecture series, and this is the final lecture series talk for this year, this academic year. We'll have another one at the beginning of the fall semester, and that's scheduled for Wednesday, October 14th. And we'll have that publicized on the STEM website at the university so you can look it up and see the entire series times. Confusingly, at first to me when I heard about it, this series focuses on stem cells, but is co-sponsored by the Center for STEM Excellence, where the two STEMs mean very different things. So stem cells are undifferentiated biological cells that can differentiate into specialized cells, while STEM disciplines are science, technology, engineering, and mathematics. So obviously they're related, but they're not the same. And if we got the botanists involved, there'd be even more STEMs. Biology seems to be full of them these days, but I hope that these introductory remarks sort of sort that out. So how did this series get started? Well, the California Institute for Regenerative Medicine, CIRM, funded my department's MA Professional Stem Cell Graduate Program through a Bridges to Stem Cell Research Grant, which is a collaboration between the UC Davis Stem Cell Program and the College of Natural Sciences and Mathematics here at Sacramento State. Our students do exciting research with UCD investigators, and we bring speakers from the UC Davis program to talk about it. Together, Sacramento State and UC Davis are bridging the causeway, though of course all of us in this room know the Hornets are better than the Aggies, right? Yeah. Before I tell you more about tonight's speaker, please take a moment to silence your cell phones. Thank you. Also, since this is such a large room, it will be difficult for Dr. Jones to field questions during his lecture. So please save up your questions until the end, and then we'll pass around a portable microphone, and you can ask your questions so that everybody can hear it. And now, after all that, let me get to the real reason you're here and formally introduce Dr. Jonas. Dr. Jonas received his MD and PhD degrees from UC Davis in 2007, and he completed his residency, clinical fellowship, and postdoctoral fellowship at Stanford in 2013. And then he joined the faculty at UC Davis in 2013 as an assistant professor of medicine in the Division of Hematology and Oncology. He believes in high quality, innovative, and compassionate care for his patients, and he focuses on adult patients, and he believes in a team-based approach to patient care and disease investigation. Among his many accomplishments, he has board certifications in hematology and internal medicine. He's a member of several professional associations and organizations. He's published numerous articles in peer-reviewed journals and is a co-investigator on a new NIH grant that supports his work. And those grants are very hard to get these days. They're harder than they used to be. So that's really quite an accomplishment. So now, with no further delays and no new definitions of STEMs, I ask you to please join me in welcoming Dr. Jonas to the podium. We're truly lucky to have him here this evening. Thank you.
Okay, can everyone hear me now? All right. Oh. Well, thank you for that really nice introduction. I appreciate the invitation to come speak with you all today. I'm just going to move this out of the way a little bit. It's a pleasure to talk to you guys today and discuss a little bit about the field of leukemia stem cells. I'm interested in both basic and translational research in this field, and I'm an active clinician. I see and I treat patients every day with acute leukemia, and I think this is a really neat opportunity, and I'm really fortunate to have the chance to actually approach it from both sides. And so today I wanted to talk about leukemia stem cells, and the title of the talk is Targeting Acute Myeloid Leukemia's Achilles Heel. So I'm hoping that by the end of the talk, we'll have had a chance to summarize a little bit about AML. I'm sure not everyone in the room is an expert. A little bit about pathogenesis and management, and also to discuss the role of leukemia stem cells as well as a new emerging part of the field, which is pre-leukemic hematopoietic stem cells, and then also talk about acute myeloid leukemia stem cell targeting therapies. So this is the outline of the talk. We'll talk some basics about AML, so you all become AML experts before the end of the day. And we'll talk about leukemia stem cells, followed by pre-leukemic stem cells, targeting, and we'll talk maybe some future directions. So AML, it's, believe it or not, a very heterogeneous disease. It's a collection of many different, they're all aggressive bone marrow cancers, and highly lethal, highly morbid. The median age is 67, so this is a disease of older patients. We'll talk a little bit about why that probably is. About 20,000 cases diagnosed every year in the United States. About 10,500 die each year, so it's a very lethal disease. It's characterized by a clonal expansion of immature myeloid cells, and this clinically leads to bone marrow failure, and this leads to infections, bleeding, and anemia. Unfortunately, it's characterized by really high rates of relapse, and this is one of the big problems in AML. These are some pictures on the right of AML blasts. So blasts are the term we use for immature cells. So AML is associated with a poor prognosis. These curves that I'm showing you here, we'll see how well the laser pointer works out. These are survival curves stratified by age. So on the left you see less than 60, on the right you see 60 or greater. 60 is the magic number clinically for AML. That's how we define older versus younger, and that's not meant to, it's just arbitrary essentially, but it does stratify the data pretty well. These curves are determined by one's genetic features, so one's cytogenetics and molecular mutations. And you can see some of the survival curves look very poor. These are, for those that are not familiar with the technique, we're obsessed with these types of curves in hematology and oncology, these Kaplan-Meier curves, these survival curves. And so this shows the proportion of patients surviving over time. So you can see over here, if you have adverse risk and you're older than 60, your chance of living 5 years is on the order of 2%. Patients who relapse or have refractory disease, this is a very big problem. You can see the survival curves are very poor. It doesn't matter how old they are. That's the great equalizer in AML. So how do we treat AML? Well, believe it or not, this regimen I'll tell you about was published in 1972. So we treat AML today the same way it was treated in 1972. I wasn't even born yet. So I think that's pretty crazy. Why is this? The disease is proven to be very difficult to treat. I think we'll learn a little bit about the understanding of the pathogenesis, which may give some clues. Maybe we just got a really good, lucky regimen back in 1972. Who knows? So the treatment is, again, defined by this age difference, 60 or above 60, and it's broken down into induction. So I'll use the term sometimes induction chemotherapy. The purpose here is to achieve a complete remission. This is how people live with AML. The next form of the therapy is called consolidation. This is to prevent relapse. I just showed you relapse is not good in AML, so the next forms of therapy are to prevent relapse. So again, we're using high-intensity chemotherapy for these younger patients, and then some patients will go on to allogeneic stem cell transplant, and that's based on this risk stratification we talked about. People older than 60, on the other hand, many of them are not candidates for this high-intensity treatment. And so some of these patients will have to go on lower-intensity therapies. Some patients are too ill to even take any therapy, and they'd go directly into hospice. Now, patients who relapse, the purpose of the goal of their therapy is what we call salvage in the leukemia world, try to get them back into remission and go to transplant. That's their only chance of being cured. It's worth noting that clinical trials are recommended for all patients with AML, regardless of where they fall into this category. So the advent of next-generation sequencing, when I was talking to the students earlier, I was sharing an anecdote that when I finished my PhD, it was about 5 years or so before I went back to the lab to do my lab postdoc, and all this stuff had happened. The next-generation sequencing boom had happened. I was still doing, Sanger sequencing was the big deal back when I was a grad student. A microarray was a Nature paper. So much was changed, and I had to learn all this language about next-generation sequencing. But it's really opened our eyes to the pathogenesis of these diseases. So this beautiful Circos plot shows the correlation between mutations and other mutations or mutual exclusivity. And these are the common mutations in AML that we see. And these ones in red are actually clinically significant. They make our way into the prognostic scoring systems. Then the Cancer Genome Atlas came out I guess 2 years ago now. This was in New England Journal 2013, and 50 AMLs had the entire genome sequence, 150 of them had exome sequencing. And they found some interesting things about AML. 13 mutations on average per genome. 5 of them were recurrent mutations, so mutations that occurred frequently per genome, so these occur frequently in the AML population. 23 genes were significantly mutated in many patients, and there was 237 genes that was mutated in more than one patient. And what I think is really fascinating about AML, and sort of interesting considering how hard it is to treat, is this is the AML mutations per megabase compared to other common cancers. AML is a relatively under-mutated cancer compared to these other cancers. I've always found that a very, very interesting finding. And I think the next figure down below might argue to why it's so hard to treat. Not this part. This just summarizes the mutations, FLT3, NPM1, DNMT3A. But I want to focus on this plot here. This is the clonal heterogeneity of AML. So we used to think AML was just a single clone. Everything was related to the same parent cell. But as it turns out, a lot of AML, over 75% of cases, had more than one clone. And this might explain why one of the reasons we have such a hard time treating it. And AML samples have these common mutational pathways, which I won't dwell on too long, other than there's a spectacular amount of data that's come out. It's really amazing. And we're still trying to mine through all this and figure out how this affects us clinically. And I think this is the clonal heterogeneity put into a really beautiful picture that was published a year before the Cancer Genome Atlas came out. You can see that when the patient has a pre-leukemic disorder, they still have a lot of clonal hematopoiesis, and then there's evolution of these clones to these daughter clones. And so by the time they present to the clinic with AML, this person has 5 different clones. So is your chemotherapy going to knock out one clone and not knock out the other 4? I mean, there's implications for this was very, very profound. So that's a little bit about the background of AML in terms of the disease, how we treat it, how we prognosticate, some of the molecular findings. I wanted to talk now for the next several minutes about leukemia stem cells. I think that's really the reason we're here today. So this is a summary, basically, of hematopoiesis, as well as a summary of the leukemia stem cell model. So hematopoiesis is driven by a hematopoietic stem cell. This is a self-renewing cell. We have these our entire lives. So they basically are able to regenerate themselves, and they spit out all the progeny that form all the downstream blood cells that you can see in this cartoon. Now, when you move down this hierarchy, the cells lose the ability to self-renew. So these cells will eventually die. They gain proliferative capabilities that allow them to form an expansion, and that way one hematopoietic stem cell can lead to millions of downstream cells. AML, we've come to realize, follows a very similar model. And this is the so-called AML stem cell model, or AML, you could say LSC model, leukemia stem cell. And you have a leukemia stem cell that has self-renewal properties. So it's a mutant cell that acquired the ability to self-renew. So it acquired stem cell properties. And it too can distribute or can form all the downstream cells of a leukemia hierarchy. It's important to note that these cells are variably defined. One of the things that was really driven home to me by my mentor when I was a trainee was that the gold standard for leukemia stem cell biology is functional assays. And that is mouse models, so xenograft models in particular. So to prove the existence of a stem cell, and it's the same with hematopoietic stem cells as is leukemia stem cells, the cells need to be transplanted into a mouse. Those cells then need to be transplanted into a second mouse, and you need to show serial engraftment. You also need to be able to show that these engrafted cells are capable of forming all the various lineages. That's the functional definition of a stem cell. Everything else is speculative. So for example, if you have a cell that has this immunophenotype, CD34 positive, CD38 negative, there's no guarantee that's a leukemia stem cell. And I think this is important. So how was this first described? Back in 1997, although there is a paper in 1994, but most people cite the 1997 paper as the seminal paper by Bonnet and Dick. And they showed that if you were to isolate from the CD34, CD38 fraction of AML cells, the 34 positive, 38 positive, these are sorted and isolated, and then the 34 positive, 38 negative are sorted and isolated. And you put these into mice, into immune-compromised mice. Well, they found that only the CD34 positive cells were capable of engraftment, specifically the 34 positive, 38 negative cells. So this is the seminal description of the leukemia stem cell. And this was their model, and it looks remarkably similar to the model I just showed you. So they hit the nail on the head right off the bat. Subsequently, it was shown that these can serially transplant. Again, remember I told you that's a key feature of leukemia stem cells. So these cells were put into a primary mouse, they were taken, they put into a secondary mouse, and then they engraft in a tertiary mouse. Here's the flow plots that show these populations, and here's quantified flow cytometry data that show that when you transplant the 34 positive, 38 negative, you get engraftment, but not with these other populations, and that's maintained through these serial passages. So this is the serial transplantation assay I'm talking about. Now, with everything in science, there's no absolutes, so challenges have come to this, what I call canonical immunophenotype of LSC. This is a paper that's very fascinating. It showed that there was two different cell populations that could transplant disease. Two different leukemia stem cell populations were identified here. One is called we now call it a LMPP, or lymphoid primed myeloid progenitor cell, and GMP, a granulocyte monocyte progenitor cell. And both of these populations, when they were sorted from many different clinical samples, led to engraftment in the mice, except for this one here. What's interesting is over here on these flow plots to the right, when you engrafted the LMPP, which is a subtype of a 34 positive, 38 negative cell, it produced 34 positive, 38 positive cells, but it also engrafted some 34 positive, 38 negative cells. When one transplanted the GMP, which is a 34 positive, 38 positive cell, it really only produced 34 positive, 38 positive cells. So both of these cells had stem cell properties, but there seemed to be a hierarchy. So one was more stem than the other one, I guess, if you want to think about it that way. So this was interesting. This study looked at different subsets of 34 positive, 38 negative, or 34 positive, 38 negative, and what they found is that this is your canonical HSC, so it forms 3% of the cells. And these other ones are not meeting the canonical HSC model. But you can see this one accounted for 34% of the stem cell activity, and they use these limiting dilution assays. And there was stem cells in the 38 positive fraction. So once again, you can identify stem cell activity in these other populations, but the frequency seems to be enhanced in the canonical one. I looked myself at therapy-related AML, which is a subtype of AML. No one's ever formally described what the stem cell is in this disease. I took 7 clinical samples, which I'm sorry they're not projecting very well, but I screened them into mice. So I did this experiment as I first saw which ones would engraft. We'll talk about the limitations of our models. The 3 of them engrafted here. So then I took these samples and I sorted out the different subpopulations, the 34 negative, the 34 positive, 38 positive, the 34 positive, 38 negative, and I transplanted them into mice. And what I found was that there was no canonical immunophenotype that predicted for leukemia stem cell activity in these therapy-related diseases. So sort of in tune with what we saw in some of the previous slides. So to summarize this whole notion of what is the leukemia stem cell, I mean, there's this canonical immunophenotype that was described by Bonet and Dick back in 1997, and it's been subsequently shown that 2 subpopulations within this overarching group have stem cell activity. And then there's the so-called non-canonical. And so, but it's been shown definitely though that the frequency of stem cells in these populations seems to exceed these populations. So it could be that there's just a matter of how much self-renewal capacity are these cells in the hierarchy retaining. And I'm totally ignoring the idea of side population and aldehyde dehydrogenase. I don't know if anyone uses those techniques for their stem cell experiments. The AML stem cell field is really focused in on the CD markers, but I just want to let you know that there's other approaches to identifying stem cells. And this is a summary of all the different populations that have shown normal hematopoietic stem cells. But once again, we have a single population here, Lin negative, 34 positive, 38 negative, 90 positive, 45 RA negative, and then a very drilled down version of the same cell where the frequency of stem cells is 1 in 10. So that's what we're after with these experiments, is what is the one that has the most activity. And the leukemia and stem cells, unfortunately, are all over the place, but maybe this is not surprising considering I just showed you this is a heterogeneous disease with heterogeneous mutations. You can imagine there's a lot of differences that could occur. And the other thing that's interesting about stem cells is the niche. Now, I could probably spend a whole different talk talking about the niche, so I'm going to condense it into one figure. But the niche is important for stem cells, and there's various models for how the stem cells interact with the niche. The model here on the left is that if I can find an arrow, okay, I'll use this one instead. In this case, the hematopoietic stem cell accumulates mutations and turns into a leukemia stem cell. That leukemia stem cell then interacts with the niche and makes the niche supportive of its growth. So that's where the stem cell affects the niche. Now, there's other models have shown that you can mutate the niche, and then this actually leads to generation of leukemia. So somehow the changes of maybe the immune milieu in the niche leads to the formation of mutations in these cells and the formation of leukemia. That's shown in the middle column. I will not talk too much about the congenital lesions on the right, but there's impact on that as well. That's probably somewhere in between. So why do I bring this up? Well, our models that we use to study stem cells, all the stuff I talked about has been done in these immune-compromised mice. It's important that we understand the limitations of these models. And so this figure on the upper right shows the history of the varying and increasing immunodeficient mice that we use, from nude mice to skid mice to nod-skid mice to what I used as NSG mice. There's also RAG mice and RAG gamma mice. And you can see that as you get more and more immune suppressed, or more and more immune deficient, I should say, you lose your B cells, and you lose NK cells, and you lose macrophage activity. But none of these have human niches. So maybe this has been a big problem all along. We've been doing all of our studies for leukemia stem cells, and we've been putting them into mice and calling that the gold standard experiment. However, the mouse doesn't even model the niche where we would normally find the stem cells in a person. So it's very possible that our understanding will continue to evolve as our mouse models continue to improve and mimic the human body more. So there's new models that are coming out. I like the name of this one, the BLT model. This one is going to hopefully mimic the bone marrow a little more. There's also cytokine mice where they're engineered to secrete human cytokines. So maybe these models will change the interaction between these leukemia stem cells we're studying and the mouse, and maybe make them behave more like in the body. Maybe there won't be so much heterogeneity in the cells. Maybe this will explain why about 40% of our samples don't engraft. Do those samples not have stem cells, or is it just the mouse model is failing us? I would argue they all have stem cells, otherwise the stem cell model doesn't make sense. So probably it's something to do with the mouse model. So maybe these models will help that sort that problem out. And so someone might say, so what? You're just going to give the patient 7+3 chemotherapy a la 1972 and hope for the best. Well, yeah, but clinically, LSC are relevant. So this was shown, someone developed a leukemia stem cell signature, used this to develop a score and a bunch of mathematics that's way over my head. I won't even come close to being able to explain how that was done. But anyway, they created this score, and they showed that it predicted who would go into remission, the event-free survival, so who would not suffer from relapse, and overall survival, so who would live. And so this is all based on gene array. So these gene changes that are characteristic of leukemia stem cells predicted outcomes. So they are indeed clinically relevant. Most recently, just a few weeks ago, there was this new paper that came out that looked at flow cytometry measuring this immunophenotypic, well, I'll call them immunophenotypic LSC. So it looked at 34 positive, 38 negative, which clinically is about the best we can do, right? We can't exactly do clinical mouse transplantation experiments in a person. So clinically, we have to rely on this immunophenotype that we use, which again has caveats. But anyway, they showed that this presence of this leukemia stem cell positivity showed a big difference in survival, whether it was measured pre or post-transplant. So you can see these curves. So people who are leukemia stem cell negative pre-transplant versus positive. And you can see the people who are cured were the people that were LSC negative. And the same thing happens if you look at the same data post-transplant. So these cells are clinically relevant. Now what about this whole idea of pre-leukemic hematopoietic stem cells? I have to admit, when I first think about this subject, I get a little bit sad. I mean, to me, this is clinically just, and you'll probably see why I feel this is challenging clinically. So again, going back to our hematopoietic stem cell model, the whole idea is that, or I should say the hypothesis here, is hematopoietic stem cells are with us our whole life, right? They're self-renewing. So these other cells that are downstream, they don't live very long, they die. So if their mutation occurs in one of these downstream cells down here, it's of no functional significance to us. That cell goes away. It's only the cells that occur in the hematopoietic stem cell that should be relevant, because those are fixed in the self-renewal process. And so that's the idea, that's the prediction, and people have set out to look at this. And I think this is very fascinating data that was just published in a New England Journal last year, presented at ASH most recently. This is showing that aging is associated with clonal hematopoiesis, with driver mutations that I showed you in those previous pictures about mutations that occur in AML. So beginning around age 40, people start to develop mutations in their hematopoietic stem cells, and this incidence increases over time. This was the one study, this is the other study. Basically, they showed the same thing, increases over time. And some of them were known drivers, some of them were drivers or were mutations that are probably drivers, but we don't know much about them. And these mutations here, DNMT3A, TET2, ASXL1, same three up here. These are all the same mutations I showed you on that Circos plot and some of the previous data. So these are mutations that are known to be very prevalent in AML. And just any old person is getting these mutations as part of the normal aging process. Probably this is why AML is a median age of 67. This is just part of the natural aging process. They showed that people that had these mutations had a much higher incidence of hematologic cancer, that's shown on the left, and people who had a higher variant allele frequency, so people whose bone marrow was more clonal, had a higher incidence of hematologic cancer. Similar data was shown in the other study. Presence of clonal hematopoiesis was associated with an increased chance of getting a hematologic cancer, and clonal hematopoiesis was associated with worse survival. So this is the model that they come up with, and it makes sense. You develop a driver mutation over time. That stem cell has an advantage. It starts to outcompete the other stem cells. Eventually, the bone marrow becomes clonal, and more mutations occur, and one gets AML. So this is an interesting piece of recent data. Some work that came out of the lab I was in at Stanford, I think, was also very fascinating and led to this development of clonal evolution model for AML. And so you start with a normal hematopoietic stem cell that's got no mutations, and over time it gradually picks up mutations, again, that it is able to fix because it's self-renewing. And then at some point, the key mutation occurs and it turns into acute leukemia. And so what's interesting about this is you have all these cells now that are pre-leukemic, and so what are we going to do about those cells when we go to treat the massive red cells up there? The other thing is this model helps to explain why I showed you earlier there's mostly oligoclonality in AML. There's usually 2, 3, 4 clones, right? Well, this model helps to explain that a little bit. You can imagine one scenario where there's a pre-leukemic cell that in parallel develops some mutations and is a subclone of the AML presentation. Or the AML clone itself has daughter clones that have acquired additional mutations, and maybe this explains the oligoclonality of AML. So how did the folks in my old lab show this? So they looked at this population here, the pre-leukemic HSC and the normal HSC. And so what they did was they took cells from an AML patient, and they sorted out normal HSC and AML cells by FACS. They also took normal T cells. T cells are long-lived in the thymus involutes when we're in high school, so T cells in adults should be long-lived that don't have the same mutation, so they're used as an internal negative control. If you try to do a skin punch biopsy of someone with AML, you're going to get a lot of AML cells. That's not going to be an optimal control. So that's why they chose this one. Then they did next-generation exome sequencing of the AML cells and the T cells, subtracted out the SNPs that were found in the T cells, considering those to be baseline. Then they went back to the individuals. They sorted individual HSCs. I think flow cytometry is just an amazing technique to me, that we can sort a single cell into a dish, and then we can do sequencing on this. Again, when I was a grad student, this isn't even unheard of. So then you can sequence a bunch of these HSC and go back and try to build together the way the leukemia developed. And this is shown graphically here. So these are 4 different patient samples, and the red cells are leukemia cells, and the blue cells are HSC. So this HSC here has a mutation that's present in the leukemia. So that's a pre-leukemic HSC. This patient, there was some HSC that had this mutation, some that had this, some that had this, and so on. So these HSC have some of these mutations. But then they didn't have some of these late mutations, like this one here, FLT3, which is known to be a proliferative driver mutation that probably occurs late. So using these data, they're able to sort of construct the formation of leukemia. But first, someone might ask, well, how do you know these are real HSC? Remember I told you earlier, how do you prove this? You put them into mice. And so they did that, and they showed that these residual normal HSC here were able to produce, there's up arrows and down arrows. These correspond to CD19 and CD33 cells. Those are B cells and myeloid cells. So in other words, they can produce bilineage hematopoiesis in the body. An AML cell doesn't produce CD19. So these are functional hematopoietic stem cells. And they contain these mutations that are present in the leukemia, but are not the driver mutation that sort of takes it over the edge to a frank leukemia. So shown in another way, this is, I don't know if it's going to project very well, but this is a mutation in TET2. And you can see almost all the cells have it, with the exception of these two HSC, these two little gray dots. This is a different TET2 mutation and an SMC1A mutation. You can see that some cells had it, some cells didn't. And then you can see this NPM1 mutation was present only in the frank leukemia cells. It was not present in these other HSC. So you can work out a model where there's some cells that have no mutations, some cells that have acquired that first hit, some cells that acquired the second and third hit, and then this additional hit with the NPM1 is what turns it into a frank leukemia. And so this is the model that they came up with for this sort of so-called pre-leukemic hematopoietic stem cells. Interestingly, so we had a nice tissue bank. I think having a tissue bank is critical to these types of experiments. That's where you take patients' cells. The leukemia is really easy. You just take some blood from someone who's at the hospital getting treatment, of course, after consent. And you can build these banks. You can viably freeze these cells in liquid nitrogen and study them 10 years later. It's great. So we do this all the time in this field. And so they looked at patients that had diagnosis samples, remission samples, and relapse samples. So sort of experimental trifecta. And they found that these mutations were prevalent in the pre-leukemic cells, typically DNA methylation and chromatin modification mutations. And then these other mutations occurred late, things like NPM1 or transcription factors, and some of the activated signaling pathways like FLT3. Shown graphically, IDH2, IDH1, DNMT3A were found in HSC, whereas for the most part, NPM1, FLT3, and RAS were not. So these are the late events. And so this model, this landscaping mutation, these IDH and DNMT3A and TET2, these are considered landscapers. They're involved in chromatin modification. They're involved in DNA methylation, epigenetics, and all that. So the mutation of these might set the landscape up for trouble later. Sorry, I was unable to get my arrow there. Set up the landscape for later, and you can see that over time they are going to proliferate. And then once you get that last important mutation, like a FLT3 or some sort of RAS mutation or something that's an activated signaling mutation, that's enough to bring it over the edge. And we usually think that that occurs not in an HSC, but in a downstream effector, which is what I showed you earlier. And then also went one step further, looked at the relapse samples. And I think this piece of work is very fascinating. So here's a patient who they had a diagnosis and a relapse sample. So the red dots are the variant allele frequency at diagnosis, and the blue are at relapse. And I want you to focus on this one, PTPN11. So this is present at diagnosis and gone at relapse. And WT1, which is not present at diagnosis and present at relapse. So this argues that there was a clonal evolution from a pre-leukemic HSC that led to the relapse in this patient. This one's a little bit different. So here we can focus on the FLT3 TKD mutation, which goes away with relapse, but RAS becomes very prevalent at the relapse. But RAS is there at the time of diagnosis. So this argues that there's a pre-existing minor clone for this AML that escaped therapy. That stem cell wasn't targeted, and then it turns into another acute leukemia. This one's a bit more obvious. All the allele frequencies match a diagnosis and a remission. So this is just you didn't get the original clone, and now the original clone becomes leukemia. So it leads to this sort of model. So you have the hematopoietic stem cells that eventually turn into leukemia, treatment, and then you can have different forms of relapse. So the dominant clone that was there at presentation, you can have a minor presenting clone, you can have further evolution of a presenting clone, or, and quite scary in my opinion, you can have progression of a clone of a normal stem cell that just was pre-leukemic. And so in clinical medicine, we talk a lot about minimal residual disease, MRD. This is what we can detect after we treat patients. So different forms of relapse can be imagined with this knowledge of pre-leukemic cells. So this is the amount of minimal residual disease, and this is time to relapse. So when there's a lot of minimal residual disease, the relapse occurs quickly, and it's likely to be a clone that was present at diagnosis. If the minimal residual disease gets knocked down way low, and then it takes a long time for a relapse, you might imagine this is more likely to be a pre-leukemic clone that acquired a new mutation, turned into a new acute leukemia. Maybe this is what we need to do to cure the disease. We need to get rid of not only the leukemia cells, but these pre-leukemic clones. That's the implication of this model and the challenge for us moving forward. Okay, so finally on to targeting these cells. How do we target leukemia stem cells? And my talk had mentioned Achilles. I don't know if everyone in the room knows the story. Probably everyone saw that Brad Pitt movie recently. But anyway, the Achilles story is this. Mother of Achilles was fearing for his safety and wanted to make him invulnerable. At least this is my understanding. I'm certainly no Greek mythology expert. So she dipped him into the river Styx. And the river Styx was meant to confer invulnerability if you bathed in its waters. So she did this. Achilles, of course, grew up and became the great warrior that we hear about at the Battle of Troy. This is him after vanquishing Hector, the son of the king of Troy. But then the other son of the king of Troy shot him in the leg with an arrow, hit him right in his, what we now call the Achilles heel. And the reason he died, so the myth goes, is that when his mother was dipping him into the river, she had to hold him by the ankle. And so that one area did not touch the water, and that was why he was vulnerable. Anyway, that's not related too much to the talk, but the idea is that maybe AML has this Achilles heel. Maybe this is a way that we can treat the cells, and maybe we can cure these diseases. Maybe these leukemic stem cells and these pre-leukemic hematopoietic stem cells are the actual Achilles heel. So there's features of these cells that can be targeted. So there's cell surface molecules, which we'll talk about several. I'll focus on that really today. I won't have time to discuss these other things, but there's abnormal signaling pathways that can be exploited. Metabolism changes are becoming more prominent now. Differentiation, maybe you can get them to differentiate into harmless, non-self-renewing cells. Leukemia stem cells have drug resistance, so maybe we can overcome this drug resistance phenomenon. And then, of course, targeting the stem cell niche. The last two, number 5, this is the approach we're taking at UC Davis with a stem cell targeting agent. And number 6, I actually have 2 clinical trials, one with a novel drug that's targeting FA3, another one that's targeting E-selectin that we're about to start treating patients with. I can't say that I developed those drugs. These are drugs that were developed by companies, and I'm having the fortune of being able to try them out clinically. So I've alluded to this before, but these are the key experiments to look at stem cells, right? You put the cells into a mouse, you look for engraftment using flow cytometry. And then you take these established xenografts and you treat them with the drug that you're interested in, and then you look at survival and engraftment. But the key is to do the secondary transplant and look at engraftment of the secondary transplant. So again, this suggests anti-LSC activity, and this proves LSC activity. I think that's an important thing to remember as we review some of these cell surface markers that have been targeted. I think this is very important work. This looks at gene expression array studies looking at HSC versus LSC, and the areas in red are expressed on LSC over HSC. And there's a lot of candidate markers. So in this talk, I was going to talk a little bit about CLL1, TIM3, CD47, which is near and dear to my heart, and CD44 as CD123. And this stuff on the side here, the pictures, the flow plots on the side show that these are really specific to AML cells and not to the normal hematopoietic stem cells. So what is TIM3? It stands for T-cell immunoglobulin mucin 3. Its role is in immune tolerance, and it's expressed on leukemia stem cell surfaces, which I just showed in the previous figure. It also is expressed in lymphoid and myeloid cells, but not in HSC. And the key feature of all these targets is that they're not in the normal hematopoietic stem cells, because if you have a drug that wipes this out, the last thing you want to do is wipe out the normal hematopoietic stem cells, because then you need to do a transplant to save the patient. So this group in Japan, they targeted these, or they isolated these cells by flow cytometry, transplanted them into mice. And so the top graph here on here is these are TIM3 positive, these are TIM3 negative. You can see only the TIM3 positive engrafted. They developed a novel TIM3 targeting therapeutic antibody. And they put this, again, the xenograft model in the mice, and they treated those mice with this antibody. And they found that this antibody led to the death of a lot of those AML cells. So as I've mentioned before, is this LSC activity? No. But it's certainly suggestive. But there was no effect on normal hematopoietic stem cells and no effect on normal bone marrow that was transplanted in these mice. So then they do the experiment that you really need to do, which is they put those cells into the mice again, they treat with the antibody, and then they do secondary transplants. And here, this is just showing what it looked like after the initial treatment. So at this stage here, you can see the AML cells are largely killed by the antibody. And this is pretty neat. So then they do the secondary transplant. There's no secondary transplantation in the mice treated with the antibody. So that basically, from a functional perspective, that wiped out the leukemia stem cells. So I think that's pretty exciting. CD44 is another important marker. This one works a little differently. This is a ubiquitously expressed marker, so we might say, oh, maybe that's not as good of a target because that's going to be on more normal cells. This has a role in cell-cell and extracellular matrix interactions. And the similar type of, you'll see it's like a broken wheel. You see the same type of experiments, and you'll see that these are all published in high-quality journals, and that's the type of experiment that's needed to convince the field. So these mice were, so they generated an anti-CD44 antibody and treated mice with AML xenografts. And interestingly here, and this is something that we deal with all the time in the experiments, is the schedule of the treatment mattered. So in this one, they got this 3 times a week, but early, and this one they got 3 times a week a little bit later. And it didn't work as well when the leukemia was more engrafted. And that's been one of the complaints about this field, is that you can sort of manipulate the engraftment conditions to make your agent look a little better. But I think I'm guessing people don't do that, but it just shows that the way the condition is set up is certainly is relevant. But importantly, it doesn't sort of matter, because when they went and did the secondary engraftment experiment shown here on the right, nothing engrafted and that was treated with their antibody. So it doesn't really matter. This is very promising. So I think this is exciting as well. It's different from the previous example. What was different about this one is it actually induced differentiation of the leukemia cells. And I can't say this is differentiation of leukemia stem cells per se, but this is differentiation of the leukemia cells. What a marvelous therapy if we could actually just not give someone chemotherapy. We could just give them something that causes differentiation of the cells into normal cells. Imagine saving the toxicity. We do this for a type of leukemia called acute promyocytic leukemia. But anyway, in this case, the antibody led to expression of CD14, which is a macrophage marker. The cells were positive by an MBT, which is a reduction assay, which is a macrophage assay. They look like macrophages and no longer look like blasts. And this is another plot showing 14. So this caused differentiation of the AML cells, which was pretty neat. CD123 is another marker. This is a marker that's actually, I would argue, furthest along in the clinic. Because at the end of the day, all of what we're doing here is to try to do something that goes into the clinic when it comes to leukemia stem cells. So CD123 is the IL-3 alpha chain, and this is overexpressed in all levels of the leukemic hierarchy. It promotes survival and proliferation. Busy figure, but basically this one here shows that and of course this group developed an antibody to CD23, and it led to impaired engraftment in many of these clinical samples, not so much this one, but all the other ones. And down here in the bottom left, there was no effect on normal cells, and there was, they confirmed that the CD123 was expressed in the leukemia cells. Now, in terms of survival of their mouse model, the survival is extended in mice that got the antibody. So that's extremely promising. So from this data, we can say this antibody targets AML cells. Now, of course, to the important experiment, which is to target leukemia stem cells, again, they did the same type of experiment I've showed many times now. And sorry, I had to take the whole figure, but I don't necessarily need to go over the whole figure. But this is the secondary transplant down here, and you can see that there is significantly impaired secondary transplant. With this is the antibody, this is the isotype control. I think what's interesting here, though, is that if you look at just the amount of leukemia cells in the mouse before the secondary transplant, you would say this isn't all that impressive. So some reason this antibody was able to wipe out those stem cells, although it didn't seem to have a huge effect on the bulk of the cells. That's the way I interpret this. And there's this homing assay, which I don't think is super relevant clinically, but it showed that it impaired the ability of the cells to go to the bone marrow and the spleen after you transplant them. So CD47, so again, I said this is near and dear to my heart. This is data generated in the lab I trained in at Stanford. And CD47 is the so-called don't eat me signal. So CD47 tells the phagocytes or the macrophages, don't eat me, I'm a good cell. And so that's basically, in a nutshell, how it works. We'll keep it simple. So you can see, and then what was found was that this was expressed in cancer cells. And it's maybe a way for the cancer cell to protect itself, upregulate the don't eat me signal, the macrophages leave it alone. This was shown here in this experiment. So when an antibody was made that targets this CD47, it leads to, this is a phagocytosis assay, so these are GFP-labeled cells. This is isotype control, 45 anti-45 antibody. With the anti-CD47 antibody, you can see the green GFP-labeled cells are now ingested by the macrophages. And down here is a quantitative, or I would argue semi-quantitative description of the same phenomenon. Because these are just based, someone is looking under a microscope and counting. I always consider that semi-quantitative, but people debate that a lot. So you can see these two different versions of the CD47 antibody increase the amount of cells that were taken up by the macrophages. Now, they did make a third one that didn't really work, so that one obviously was pushed to the wayside early. Now, the same antibody could eliminate AML cells that were engrafted in a xenograft. So again, we're marching through this, right? We figured out how the antibody works. Now we're targeting AML cells inside of the xenograft. And of course, we still need to prove stem cell activity later. So on the right here, you can show quantitatively there's a decrease in engraftment from the left to the right. That's isotype control versus the antibody. And the bone marrow shows, this is mice that got the isotype control. So these are heavily engrafted with AML. These are mice that got the antibody. Their bone marrow has been cleared out of these disease cells. This is sort of interesting. These are two mice that the bone marrow didn't clear. But what you can see are in those little white patches with the black arrows that don't probably protect very well. These are areas with macrophages that are actually eating the cells. So they managed to capture some functional images of what was going on. So then the next experiment that we talked about that's logical is to see if it affects LSC. And so on the left, this is what I would say the sort of silver standard experiment, but they measured the immunophenotype, right? 34 positive cells. Were they impaired by this antibody? And you can see here that there's fewer CD34 positive cells after antibody treatment. Again, that's suggestive but not proving. On the right, this is the secondary transplant experiment. And you can see in the presence of the antibody, nothing was engrafting. So this antibody's got AML activity, it's got stem cell activity, and it works by impairing this don't eat me signal. So at least to this model. So they have the hematopoietic stem cell on the left in the blue that's got CD47, interacts with SIRP alpha, which is the receptor for CD47 on the macrophages. And it's not eaten. Well, the leukemia stem cell has a pro-phagocytic stimulus, which has been subsequently found to be calreticulin. Calreticulin interacts with this receptor on the macrophage, and it's an eat me signal. But the AML cell upregulates its CD47 to counteract this. So it upregulates that don't eat me signal to counteract the eat me signal. Now what happens if you give this antibody? Well, you block this interaction. A normal stem cell isn't bothered by this because it doesn't express the eat me signal. And then whereas the leukemia stem cell does, and so now this calreticulin signal is active and the cell is ingested by the macrophage. So that's the theory behind how this works. The last thing I want to talk about from a targeting perspective is CLL1, and this is what we're working on at UC Davis. So this is a C-type lectin-like molecule 1, and that's the name for it. It's actually not really known what it does. It's just something that's been identified as being on stem cells. It's really not expressed in any adult tissues. That's a good marker for a stem cell targeting agent. That's good. It's expressed in 85% or more of AML samples, so check, right? You have a marker that's not expressed much in normal cells, and it's expressed in 85% of leukemias. That's good. This is just further evidence that it's expressed on leukemia cells and not on normal bone marrow. So the one on the left is the AML sample, and the ones on the right are normal bone marrow or peripheral blood. And when you put it in, when you sort for CLL1 positive cells and put them into xenografts, they engraft. So it seems like it's a stem cell marker. A CLL1 antibody was developed. This antibody has CDC activity, complement-dependent cytotoxicity activity. It's got antibody-dependent cellular cytotoxicity activity, so that's ADCC. So that's what these figures on the left show. The top one shows CDC, the bottom one shows ADCC. So these are two different versions of a CLL1 antibody. This is isotype control. This is the relevant antibody. This is isotype control. So this antibody works both by direct killing of the cell, but also by recruitment of immune cells. And you can see, I think this is projecting well enough, but what's really fascinating about CLL1 is it's internalized after it binds to the antibody. So at 0 minutes, it's on the surface of the cell. At 30 minutes, you start to see it inside of the cell. And at 120 minutes, it's fully inside of the cell. This was confirmed with a protease assay that's done at different times, and this is no protease, and this is various times that the antibody is exposed to the cell, then protease is given. And the longer it is, the more antibody is inside the cell. What a great target. Now you have something that's expressed only on stem cell, or leukemia stem cells. It's not expressed much in other tissues. It's also expressed on the blast, the bulk cell, so it's actually expressed on all the AML cells. And it's internalized after you bind it. So what a way to maybe deliver something inside of the cell. Now this antibody wasn't really a hit when it comes to killing the cells. This is a AML cell line xenograft model, so not patient-derived, but cell line-derived. And this is isotype control, and in the presence of the antibodies, there was some impairment of the growth. But I would argue we've certainly seen better examples on previous slides of things that were much more dramatic. But the advantages I just mentioned, so it's a LSC and blast marker, it's high penetrance in AML, and it's internalized. A disadvantage of an antibody, antibodies have half-lives measured in weeks. So if this, one thing I didn't say is that this CLL1 has been found on progenitor cells. So you might wipe those out with an antibody that's around for weeks. But if you could develop something that's not around for very long, those could be, the person would recover from that, their stem cells would make more progenitor cells, and they'd be fine. And we've also known that high-dose chemotherapy can cure some people with AML. That's what we've been doing since 1972. So clearly there are some people whose AML can be cured with high-dose chemotherapy. The trouble is we can't give high-dose chemotherapy to most people. It's too toxic. So our hypothesis is that a short-lived CLL1 targeting nanoparticle could deliver a high-dose chemotherapy payload directly to AML blasts and LSC and decrease systemic toxicity. Before I joined the group at Davis, they had done a phage peptide library display, and they had isolated different peptides that bind to CLL1. These are all the various candidates. They ended up choosing for further study the second one here because of the change from the proline to the aspartic acid made it more hydrophilic, easier to work with. They showed that this CLL1 ligand could bind to cells that express CLL1. Well, that's perfect. That's what you want to see. You can see over here, this is a cell line that doesn't express CLL1, but it's now expressing it because it's got a retrovirus added to it that has CLL1 in it. And it's positive in the setting of the ligand. And these are also the same type of experiments using a biotinylated peptide, biotinylated peptide. These are two cell lines that don't normally have CLL1, but are engineered to express it. And you can see that it's binding. So it binds to the target we wanted it to bind to. And so Dr. Lamb at UC Davis has pioneered this nanomicelle technology. And what he does is he takes these chemical structures called telodendrimers. These are PEG, they have PEG at the end, and then they have a oligomer of cholic acid on the other end. And these things self-assemble into these nanomicelle structures. And you can actually, I guess I'll move on to this. You can actually have drug that gets incorporated inside of this structure when these things self-assemble. So that's what it would look like, or a little bit more pretty on this one. And so these are the various telodendrimers that have self-assembled with the drug inside of it. And to the end of the telodendrimers, you've chemically added your binding target, your CLL1 ligand. So this is the general drug that we've developed. So the advantages are you could deliver a high-dose drug or a combination of drug directly into whatever cell this thing binds to. In this case, we think the LSC. You could also test various combinations in the drugs. I think one thing that could be fascinating is testing things like siRNA or other non-chemotherapeutic agents inside of these nanomicelles. So the sort of limitless possibilities, right, of what you could put inside of these things. I shouldn't say limitless because there is hydrophobicity that does affect what you can put in. But that being said, there's a lot of interesting things you can do. There's the theory that these things will break down as the lipids are constantly taken away as it goes through the circulation. So some of the drug will release into the body over time. So it might increase the area under the curve. And this might mitigate the toxicity. Anyway, these are the advantages and the theories that we've tested out to prove. Now, this is two different imaging techniques that show the nanometer scale of these particles. So they really are nanoparticles. This is a figure showing that the particle gets inside of CLL1 expressing cells. That's inside the nucleus. In this case, it's in the cytoplasm. This is a nanoparticle with a dye, and it's being cells that have CLL1 are being exposed to this nanoparticle with a dye, and it's increased in the targeting one, and the non-targeting one has a little bit, but doesn't really get in the cells as much. And you can see here daunorubicin, which is the chemotherapy drug we're interested in, it's also fluorescent, so you can actually directly measure it. So in a CLL1 expressing line, daunorubicin is preferentially taken up in the setting of this targeting nanomicelle as opposed to just a non-targeting nanomicelle. Now this is AML blast on the left, normal blood cells, and hematopoietic progenitor cells. And so the only, of these flow plots, the only thing that took up daunorubicin was the leukemia sample that was treated with the targeting nanomicelle. So it seems to be specific for these leukemia cells. This is a cell culture experiment, but without focusing too much on the numbers, this is the targeting nanomicelle. These are normal blood cells, and these are what, I guess, probably better, we should say CD34 positive, 38 negative cells are not functionally defined LSC. But in vitro, they do kill, these do kill the LSC preferentially over the free daunorubicin, and that is statistically significant. Taking mice and putting a cell line model into them, this is also sensitive to these nanomicelles. So this is the free daunorubicin arm. This is no treatment. You can see they die quickly. And then this is the survival is extended with this drug. So this is quite promising. And so we've done some pharmacokinetic studies in mice and rats. So we're on the sort of various steps towards trying to get this into a patient. We have to do all the mouse modeling and all the animal modeling before we're allowed to try it out in people. And so this is the PK studies in mice and in rats, and the area under the curve with the nanomicelle formulation is like 30-fold more than the free drug. The free drug is the concentration goes up rapidly in the bloodstream, and then the half-life is minutes. Now, the interesting thing about the nanomicelle that will be, I don't know, I think this could be one challenge down the line and maybe something that needs to be worked on, is it does seem to accumulate in the liver, the spleen, and the kidney. So these are the reticuloendothelial system, right? The parts of the body where lipids and various things get trapped in these organs. So I don't know. It's going to be interesting to see whether this translates into anything. It hasn't in the mouse, but whether or not it could translate into humans as a problem remains to be seen. So the big problem with daunorubicin is actually not anything to do with the liver, the kidney, or the spleen. It's cardiac toxicity. The reason I can't give my patients unlimited amounts of daunorubicin is because I will put them into heart failure, and they'll end up having more problems from their heart failure than they will from their leukemia. And so, but we do know also that if I give those doses of daunorubicin, people do better. So there's our problem. Can we get those daunorubicin doses into patients, but spare their heart? So with the nanomicelle targeting formulation compared to the free daunorubicin, if we just give the mice a very high dose of the daunorubicin, and then targeting nanomicelle formulation, there's really no effect on the cardiac myocytes. And you'll have to take my word for it, but this is basically a bunch of necrotic cardiac tissue. So basically the free daunorubicin at that same dose is basically really done a number to the cardiac tissue, whereas the targeting nanomicelles did not. Even a lower dose of the nanomicelles, there are some, I don't know how well it shows up, but there are some apoptotic cells here, and you don't really see any in the targeted nanomicelle. Troponin is another clinical thing that we use, another clinical lab that we use to look at heart damage. So it's been shown that if you look at troponin levels right after chemotherapy is given, that can predict who's going to develop heart failure 5 years later if they happen to be cured. And so looking at the troponin level in the mice, the nanomicelle did not cause a significant increase in the troponin levels, but the free daunorubicin did. You can see the free daunorubicin. And so it protects the heart. This is a caspase assay looking at cardiac tissue. Again, the same finding, the free daunorubicin and the nanomicelle daunorubicin. So this is doing what we were hoping, which was to mitigate the toxicity of this treatment and allow us to give a higher dose of the treatment to the mice. So now we're really sort of moving into the PDX models. I said before, we've looked at a cell line model, which I think is not as ideal as looking at the PDXs. So we've generated a PDX model, as I described before. We put the human sample inside of into the mouse via tail vein injection, wait for it to engraft. So this is the engraftment of the human cells. This is using HLA. Mice don't have HLA. They have this H2. They have different, it is the mouse HLA. It's just not called that. But this antibody doesn't cross-react with the mouse. So these are going to be human cells, and this is CLL1. So you can see the engraftment is with mostly CLL1 positive human cells. On the right, you can see that there's some mouse cells, and they do actually seem to have some CLL1 expression, so I don't fully explain that. I can't fully explain that. It might be that the antibody we're using for flow is sticky. There could be some cross-reactivity between human and mouse CLL1. I'm not totally sure. Anyway, we treated these established xenografts with our drug for 3 days consecutively, which is how we treat humans, and we performed analysis after about 4 weeks. We also took some mice and did secondary transplants, and unfortunately I don't have the data yet from this experiment because it's actually ongoing. 2 weeks from the time of treatment, in our treatment arm, after these 3 doses, there was a decrease in the level of engraftment compared to free daunorubicin or PBS control in actually both the non-targeting, which is what we were surprised to see, as well as the targeting drug. But by 4 weeks, the effect was best in the targeting drug. That's the one on the far right there. And it didn't seem to affect the size of the spleen all that much, although there was a statistically significant difference in the size of the spleen in the targeting nanomicelle. So I would leave this particular slide by saying our drug is promising. We have not proven that it's anti-LSD activity yet, though, I would say. And maybe like the other antibodies that didn't have a total wipeout of the AML, but it totally wiped out the LSD. Maybe that's what we're going to see when we get our secondary transplant back. Anyway, I'm just speculating, but I think it's very promising. There might be some optimization that needs to be done. So I hope I've shown you, I don't know how I'm doing for time, but I hope I've shown you that AML is a devastating and biologically heterogeneous disease. I think we could all agree on that. It's initiated and maintained by these rare, self-renewing leukemia stem cells that are resistant to therapy. It's caused by the stepwise accumulation of mutations in these pre-leukemic hematopoietic stem cells that serve as a reservoir for relapse. Makes our life in the clinic a lot harder because of these cells. Elimination of these LSC, and I would argue probably these pre-leukemic HSC, is probably likely to improve outcomes in AML. Probably that's maybe one of the ways that we can cure this disease. It's probably not the only way. But this might be the Achilles heel of AML. We can somehow get these cells targeted. And I showed you some of the weaknesses that we can exploit with new drugs in these stem cells. So obviously there's more targets out there. I gave you that whole list that obviously I wouldn't have time to discuss. There's metabolic markers. There's these differentiation signals. So there's a lot of other things that need to be evaluated to see which is maybe the most promising things. I think this is another exciting area of clinical oncology and hematology, and I think I'm sure very few people in this room haven't heard about the big boom in immunotherapy now for cancer. So immunotherapy might be a big strategy to target leukemia stem cells. So those markers I told you about, CLL1, CD123, so there's already been development of these things called bispecific T-cell engager peptides. I think these are fascinating. These are like two parts of an antibody that are just made in one peptide. And it takes a CD3 cell, a cytotoxic CD3 cell, and it brings it directly to the cancer cell where it can eradicate the cancer cell. And so these have been developed for both CLL1 and for CD123, and CD123 is pretty close to the clinic. The other thing that's had a lot of popular press lately is the chimeric antigen receptor T cells, or so-called CAR T cells. These are when you take the cells out of someone's body, you engineer them in vitro and put them back in the patient. And you basically engineer these T cells to attack a marker that's on the leukemia. And they've shown the ability to cure childhood ALL with this approach. So we might, and actually CD123 it's starting to get into the clinic with these CAR T cells as well. So this ultimately might be better than all of our other approaches that we've tried so far. I think all this stuff is very exciting. But again, it's still utilizing these vulnerabilities of AML stem cells, right? These things that are expressed on the stem cells that are not expressed on the normal stem cells. I mean, imagine if you had CAR T cells, a bunch of activated cytotoxic T cells that go into a patient and are wiping out their normal stem cells as well. That would be a big problem. You need to transplant a patient or else they're in trouble. The other thing that's got a lot of press lately is called program death 1 and program death 1 ligand, so PD1 and PD1 ligand. Antibodies targeting these proteins have revolutionized the care of melanoma. People are surviving with metastatic melanoma in ways that they never did before. And this is a way that cells turn off the immune system so they can escape immune attack. So the LSC are likely to be using these pathways too. So there are phase 1 clinical trials now with some of the drugs I've talked about in this presentation. We don't have any yet at UC Davis, but Stanford has opened the CD47 trial for solid tumors, and CD123 antibody trials are underway. And we have our new R01 funding to further the project I described. We're going to try to optimize our nanomicelle further and do a lot more of the in vivo work that's necessary before we can really say it's stem cell targeting. And we still need to do more animal studies in order to get it into the clinic. So we're looking forward to that. And like everyone, you need good mentors to get it to move ahead in science. I'm particularly grateful to Dr. Majetti, who was my postdoctoral fellowship mentor at Stanford. He taught me the leukemia ropes and the stem cell ropes. He taught me to be very cautious about what I call a stem cell. Dr. Pan, Dr. Henderson on the left are my colleagues at UC Davis that we're working together with on the CLL1 project. Dr. Lamb is not pictured, but he's also part of the group. This is our lab up in the middle. I'm also a holder of a UC Davis Paul Calabrese Clinical Oncology K-12 award. So this is an NIH program. And Dr. Pan, Dr. Henderson are my mentors. And these are some of my current and past funding sources. And to close, I think it's sort of fun. When I was a grad student, my mentor told me about his academic lineage, and I sort of thought, okay, maybe I'll trace my academic lineage and make it analogous to a hematopoietic hierarchy. So I'm lucky to be a grandson of Irv Weissman, who's probably pretty well known in this room. He's had a big role in the development of these models that we're showing today. He trained Ravi trained me, and now I'm lucky enough to train some downstream macrophages and granulocytes and platelets. So with that, I'd like to take questions.
So I'm curious, is your nanomicelle targeting the CLL1?
Yeah.
If you're able to successfully target that, do you add a selective pressure for those stem cells not to be expressing that marker? And do we run the risk of losing that marker?
I think that's definitely possible. And that's been, the CAR T cells I was just talking about. They are not 100% universal, and they've now demonstrated that when the ALL relapses in these CAR T cell treatments, they relapse without CD19, which is the CAR T cell target. So AML is a very smart disease, despite it's not heavily mutated, but it's still very smart. And so you're right, there probably is going to be some mechanism of escape. I think combinations of drugs are going to be important, because if we get the cells in, if we get the target, the chemo into the cells, And we can maybe eliminate them before they have a chance to, not express the CLL1 ligand. The other thing that's interesting is you saw there that some of the nanomicelles go into cells non-specifically. So it might be possible that we'll get the target into the leukemia stem cells that don't have CLL1 on them. Yeah.
And is gene editing at all part of this discussion yet?
Not so much yet, no.
Okay.
Hi.
My name is Taisia. I had a quick question. So, and this is general, but going back to the beginning of what you said, I know you mentioned alterations in models of mice, which could better accommodate reflecting the nature of human cells and contributing to the study. So, would you say that other living organisms could serve as a better option, or do mice serve dominance over other options for reasons more than just being abundant? Why?
Yeah, that's an interesting question. I think I've never used this model, but zebrafish are another common model used in AML, in hematopoiesis studies. And I think part of the attractiveness of the model is that you have to find something that will take the human sample and have it engraft. So zebrafish, I've not seen actually human AML cells engrafted in them, but I've seen a lot of studies on normal hematopoiesis. And so I guess in the short, the short of it is that the immune suppression has to be so profound in these animals that we have to have that. And I think that's why the mice make such a nice model. It's hard to imagine that we would have such a profoundly immune deficient primate or something like that. It'd be a little harder to do that. It's feasible, but it's not something that, and the cost of the mice is also an attractive feature. So I would say right now there's certainly not much in the field that's outside of the mouse, but I wouldn't say that means that there couldn't be. But I think these other features like cost and the ability to engraft the cells in these other organisms could play a role. There was a fascinating Cell paper that came out this last week that raises the possibility of clams being a model for AML. I thought that was really neat. So there's this. So look at Cell if you have some free time. It's a really neat paper. They showed horizontal transmission of clonal AML among clams.
So you wouldn't find the mice being limiting?
Pardon?
You wouldn't find the mice as a model to be limiting. It's just.
Well, they're limiting for the reasons I said. But if we can engineer them to have human bone marrow and microenvironment, then I think that would really overcome a lot of this issue. One of the things I didn't show today, which is fascinating, is that someone in my lab, he's actually making bone marrow sort of three-dimensional models inside, like under the skin of the mouse. People do this all the time with things like Matrigel and stuff. But he's using platelet-rich plasma from patients, which have a bunch of growth factors and so on, and using it to create these little balls of bone marrow inside of them, under the skin of the mouse. Then you can take a needle, puncture it into there, and inject a few stem cells, and you have your own little mini human bone marrow cavity. And so models like that might really overcome some of these problems. But I think part of the issue is this immune regulation, and I think it's going to be a challenge for studying all these immunotherapies we're talking about too. And I think there's still a big role for mouse leukemia models, not just human xenografts too. And I've been interested in talking to someone at the vet school, and I have not done this yet, but about talking about some of the leukemia. There's feline leukemia models, for example, and some of the transplant questions that we can ask. So. I don't know. It's a long-winded answer to your question, but I think right now the mice are the best model we have.
Okay, awesome. Thank you.
I was really impressed by that drug delivery system. Is that being at all used in research yet, or is that something you said that came out of your lab? But is that expanding to other areas?
The nanomicelle? Yeah, we have these meetings that we do once a month. We call them innovation group meetings. And we come up with all these neat ways that we can use these nanomicelles. So put prednisone inside of them and inject them into the CSF to treat CNS tumors. Could we target them with, it's actually been targeted, one of the cardiologists at UC Davis is targeting them for oral inhalation to go to the lungs of people with asthma to deliver asthma drugs. And so there's all sorts of ways that you can, change this drug delivery system to tailor your need. You can put whatever you want on the end of these things, any target you can imagine. And you can almost put whatever you want inside of them. I think that's what makes them really, really exciting. And this idea of using blasting the AML cells with higher doses of chemo, maybe that's not going to be the best approach in the long run. Maybe an siRNA or something. So I think this is a really neat platform. And I'm hoping that we can have tons of clinical trials eventually, because Dr. Lam has just developed this really great technique. He needs the clinical people to think about how to use it. Oh, thank you.
Well, we would like to thank you for joining us this evening,
Thank
and we
you.
look forward to seeing you next semester at the beginning of the series. And thank you for coming, and we'd like to present you with this large frame of your poster
Thank you so
for
much.
your presentation.
Thank you. Thank you very much.